Hello, pharmacists, pharmacy technicians, and other healthcare providers. Welcome to APHA's inaugural episode of the 15 on COVID-19 series. Today we'll be talking about antimalarials as antivirals. My name is Dan Zlott. I'm the Vice President of Education at APHA. Prior to joining APHA, I worked at the National Institutes of Health as a clinical pharmacy specialist in immunotherapy, oncology, and immunology. Thank you very much for joining us today. I have nothing to disclose. The learning objectives for today, at the end of this presentation, you should be able to describe the hypothesized mechanisms of quinolone antimalarials in the treatment of COVID-19. So just really briefly, what is the 15 on 19 series? Well, it's a regular education series that will provide education about the various aspects of COVID-19 disease, help you separate hypothesis from rumor or even outright fiction, and provide 15 minutes of CE for pharmacists and pharmacy technicians. So what are the ground rules for this series? Well, episodes will be 15 to 20 minutes in length so that you can quickly get the information that you need. We're going to cover one topic in each episode, and we'll attempt to clearly state whether each idea or concept is fact, hypothesis, or rumor or fiction. That way you can have a better sense of what's out there in terms of just trying to evaluate the incredible amount of information that's coming out about COVID-19. It goes without saying, information on COVID-19 is changing daily, so what's true today may not be true tomorrow. What's hypothesis today may be fact tomorrow, and what's rumor or fiction today will probably still be rumor or fiction tomorrow, but we'll still be checking just in case. Now, that said, it's your responsibility to stay informed. We'll do our best to update information that becomes outdated, but we can't guarantee the accuracy of information past the date of presentation. Additionally, have to have a disclaimer in there. Information in these presentations are the opinions of the speakers only and do not necessarily reflect the opinions or guidance of the American Pharmacists Association. So we've all been hearing a lot on the news and just about from every media source out there about chloroquine and hydroxychloroquine for the treatment of COVID-19 disease. So the first time you heard that, you might have wondered, why are we talking about antimalarials as a treatment for a viral disease? Well, let's dive in a little bit and try to separate fact from fiction here. So this is a fact. Believe it or not, people have been researching the utility of antimalarials for the treatment of various viral infections, including HIV and coronaviruses, for more than 20 years. Another fact, the outbreak of SARS in the early 2000s, 2002, 2003, depending on which piece of literature you read, was also caused by a coronavirus. In this case, it's the SARS-associated coronavirus, or SARS-CoV. Now, this is a different strain of coronavirus than the coronavirus, which is SARS-CoV-2, that's responsible for COVID-19 disease. One last fact, the SARS outbreak in the early 2000s spurred research into various compounds that might be effective for the treatment of SARS. And one of the compounds or family of compounds that showed great promise was chloroquine. So both chloroquine and hydroxychloroquine had various levels of efficacy. And so we'll talk about that in the next couple of slides. So you might be wondering, how exactly does an antimalarial treat a viral infection? Well, let's go through some of the proposed mechanisms of action, and then once we kind of give an overview, we'll go a little bit deeper and explore these mechanisms in more detail. So the first one I'm going to call both fact and hypothesis, and that's that chloroquine inhibits viral replication, and it does that through a couple of different mechanisms. The first is the inhibition of coronavirus entry into the cells through endocytosis. The second is through inhibition of pH-dependent steps in viral replication. And thirdly, chloroquine has been shown to inhibit the MAP kinase pathway, which plays an important role in coronavirus replication. So another potential mechanism, and this one I'm going to call hypothesis, is through the inhibition of immune-mediated organ damage. And that's something we saw a lot early on with SARS, and it's something that the data is also showing with COVID-19. So let's dive a little bit more deeply now into these mechanisms of action. So let's start with the first one, inhibition of viral endocytosis. So in other words, entry of the virus into target cells. So before we do that, it might be helpful to review just the whole process of viral replication. So first off, the virus interacts with the cell, typically through some type of a fusion protein. It gains access to the cell. Once it enters the cell, it typically is contained in some type of a lysosome, or it encounters lysosomes. And from there, those lysosomes are very pH dependent. They help to break down the coating on the virus. And so we call that virus uncoating. That releases the infectious genetic material into the cytoplasm of the cell. From there, the cellular machinery is able to transcribe and also translate any of the infectious genetic material and start producing viral components of the virus itself, whether that's replicating the viral DNA or associated viral proteins. From there, it undergoes post-translational processing of envelope glycoproteins of some of the proteins themselves as they get maybe chaperoned and kind of formed into different groups. That takes place in the endoplasmic reticulum and in Golgi bodies. And then from there, they bud both out of the Golgi bodies or even out of the cell. And then you have a mature virion, and that virus particle can then go and infect other cells. So that's just the overall process of viral replication, and that's what that diagram shows. So how does chloroquine or hydroxychloroquine inhibit viral endocytosis? Well, it turns out that SARS, the original SARS, and actually this was recently discovered just a few days ago, the COVID-19 version of the SARS virus binds to angiotensin converting enzyme 2 on cell surfaces and then is endocytosed into the cell. So it's interesting, chloroquine appears to alter the glycosylation of ACE2. So it doesn't really change the protein structure itself. What it changes are all those post-translational modifications of the ACE2 protein. And what we think this does is it makes the interaction between the SARS virus and ACE2 less efficient, and this slows down and may even inhibit viral entry into the cell. So if the virus can't get in, it can infect the cell, and at that point it's pretty well neutralized. Now, this data is based on the SARS virus, the original SARS virus. It's an extrapolation to assume that the same is true of the SARS-CoV-2 virus that causes COVID-19. We do know that, as I mentioned earlier, the SARS-CoV-2 virus does bind to ACE2, but we don't know whether it's true that chloroquine and hydroxychloroquine prevent the interaction between the receptors on the virus and the ACE2 protein. So now let's talk about some of the other mechanisms, and that's the inhibition of pH-dependent steps in viral replication. So chloroquine is an interesting drug. It's a weak base, but it's able to get across cell membranes pretty readily. We won't talk about Henderson-Hasselbalch and all that kind of stuff, but throw you back to that if you're interested in learning more about that. But what it does is it tends to actually get concentrated in acidic vessels within the cell, and because it's a weak base, it actually increases the pH. Now keep in mind that almost all proteins in our body are exquisitely sensitive to pH. So if you change the pH just a little bit, they stop functioning as they were intended. And in this case, that's a good thing for us. We think that this potentially disrupts multiple steps in the viral replication pathway, including viral uncoating. So I mentioned earlier that once the virus is actually inside the cell, it typically interacts with a lysosome. Lysosomes are very acidic, and that acidic environment helps to break down the coating of the virus and release the infectious genetic material within the virion particle. So if you, again, even if the virus gets in, but it can't uncoat itself, that infectious material never gets released, and the infection is essentially halted at that point. Additionally, it may interfere with post-translational modifications of viral proteins. So once all the genetic material has been transcribed, and once any proteins have been translated into proteins, all that stuff goes into the endoplasmic reticulum and the Golgi body for post-translational modification, glycosylation, pulling together of different proteins, etc. So it turns out that that's also a pH-dependent process, and so by inhibiting the interaction within the endoplasmic reticulum and the Golgi body, we also inhibit the ability of the virus to package itself. Lastly, it turns out that there are certain proteins on coronaviruses that are responsible for their ability to bud out of the Golgi complex and basically form mature virion particles. And so chloroquine has been shown to inhibit the ability of coronavirus to bud out of the Golgi complex. And of course, if you can't get out, again, you can't produce mature virion particles and the infection is effectively halted. Now, the last proposed pathway in terms of inhibiting viral replication is around the inhibition of MAP kinase. It turns out that coronaviruses are very effective at upregulating or increasing the activity of the MAP kinase pathway. Broadly speaking, the MAP kinase pathway controls a lot of cellular function. It basically is sort of one of the key points of cell replication. It turns on all the cellular machinery that our cells use to replicate themselves. And so coronavirus takes advantage of that. So it's been demonstrated in multiple studies that inhibition of the MAP kinase pathway has been shown to inhibit viral replication, and it turns out that chloroquine has been shown to inhibit MAP kinase activity, as well as to inhibit coronavirus viral replication. So let's move on now to another area that's hypothesis. And that is the inhibition of immune-mediated organ damage. So the image here is actually of COVID-19 patients, and if you've looked at CT scans, you can see that these are pretty gnarly-looking CT scans, an incredible amount of infiltrations, particularly in panels B, C, and D. And this is very common with what you see in COVID-19. Our current estimates are that a lot of this whiteout effect is actually due to an immune response within the lung tissue. A lot of that inflammation is caused by the immune response, not necessarily by the virus itself. Well, it turns out that chloroquine and hydroxychloroquine are potent immunosuppressants, and if you think about some of the things you may have seen them prescribed for, they're used for a variety of autoimmune disorders, including SLE, systemic lupus erythematosus, rheumatoid arthritis, other autoimmune disorders. So when you look at this in the lab, it's pretty interesting. Chloroquine has been shown to inhibit the production of a variety of pro-inflammatory cytokines, and these include interleukins, interferons, and tumor necrosis factor alpha. Now, what's interesting about this, if you think about some of the more recent anti-rheumatologic treatments that are available, particularly a lot of the monoclonal antibodies, the interleukins and the tumor necrosis factor alpha family in particular are targets of some of those newer drugs that are designed to tune down or turn off some of the immune response in autoimmune disorders. And when you think about interferons, prior to the release of the most recent group of hepatitis C drugs, interferons were really drugs that we used in an attempt to upregulate or initiate immune activity in an attempt to allow our bodies or our immune systems to help clear hepatitis viral infections. So it's kind of interesting that the way that we're modifying the immune system may also have some interesting tools that we can use for COVID-19. More on that maybe in a future episode. So now, so those are all hypothetical, right? The reason I call some of those fact and hypothesis is that all those tests were done in the lab, and that's great. But one thing we know for sure is that what happens in the lab doesn't always replicate our experience in actual humans when you go to treat disease. So the fact is that, yes, we can demonstrate that in a petri dish, in a viral culture, we can downregulate viral replication, inhibit viral replication. We can downregulate some of the cytokines that we know cause autoimmunity. However, there's not necessarily a lot of clinical evidence to back up the fact that those things happen in human beings who are infected with SARS, or in this case with COVID-19, with the COVID-19 virus. So what clinical evidence is out there to actually back up the use of these drugs? So let's dive into that. So there are a ton of ongoing trials, as you might imagine. To date, really only 2 have reported results that I'm aware of. One article summarizes the Chinese experience from multiple medical centers across multiple studies, and they reported, I'm going to quote this directly, they reported that results from more than 100 patients have demonstrated that chloroquine phosphate is superior to the control treatment in inhibiting the exacerbation of pneumonia, improving lung imaging findings, promoting a virus-negative conversion, and shortening the disease course. And the reference is there for you if you'd like to look at it yourself. It's a 2-page article. Unfortunately, there's really no additional detail provided here whatsoever regarding study design, patient selection criteria, outcomes, safety, etc. And so I would call this exceptionally poor quality data, which really doesn't support the recommendation one way or the other of the use of chloroquine or hydroxychloroquine at this time. So there's a second study that's recently been released, and I think a lot of the news that we've been hearing is largely based on the findings from this study. So let's dive in and explore this one a little bit more. This study provided much more detail, but still has significant limitations. So this study was a multi-center, multinational, open-label, non-randomized trial comparing hydroxychloroquine or hydroxychloroquine plus azithromycin, and that was added at the author's discretion, against control patients who received best available care. So already we can see some significant limitations. It's an open-label, non-randomized trial. There's some oddities there with the addition of azithromycin at the investigator's discretion. There really wasn't a set protocol for how and when that was added. But again, it's what we've got. Additionally, all the hydroxychloroquine patients were treated in one center. Patients from all other participating centers served as controls, as did patients who refused hydroxychloroquine from the original center. And this study largely took place in France, and I believe there was one additional center in Vietnam, if memory serves. So let's start off with the results. So in this figure, this is figure 1 from the paper, we see the control group in black compared against the hydroxychloroquine group in red. The vertical axis is the percentage of patients with PCR positive samples. So in other words, that's the percentage of patients where they could still detect the viral RNA or viral genetic material. And this looks pretty impressive. You can see that there's clear separation between these two lines, and the results become statistically significant, day 3, day 4. And so this looks really good. However, keep in mind this is a very small n, and we'll go into some of the other limitations as we go through some of the additional details about the study. So here's another chart looking at the results. And so this one also compared controls to hydroxychloroquine patients. So the controls are in black, hydroxychloroquine patients are in blue. And because they added azithromycin to some patients, those patients were reflected in green. And here, interestingly enough, it shows that patients who got the combination of hydroxychloroquine and azithromycin were all viral PCR negative by day 5 or 6, which sounds very impressive until you realize that there's only 6 patients in that arm. And again, this was added at the investigator's discretion. There really was no preset criteria for that. And the reason the authors gave for adding that was to prevent bacterial superinfection in patients who were infected with COVID-19. So let's dive into this a little bit more and explore some of their study design. So who are these patients? These were basically hospitalized patients who were older than 12 years old who had a PCR documented SARS-CoV carriage in their nasopharyngeal sample at the time of admission, and they were included regardless of clinical status. So pretty broad inclusion criteria. Exclusion criteria were equally open. Known allergy to chloroquine or hydroxychloroquine, known contraindications to chloroquine or hydroxychloroquine, including retinopathy, G6PD deficiency, QT prolongation, things we'd usually worry about when treating patients with hydroxychloroquine. And then lastly, breastfeeding or pregnant patients were also excluded. So who are the patients? Again, a total of 42 patients were enrolled. So this is a very, very small study. 26 patients received hydroxychloroquine, and of those 26, 6 patients were lost to follow-up. 3 patients were transferred to the ICU, 1 patient died, 1 patient left the hospital, and 1 patient stopped treatment with hydroxychloroquine due to nausea. All told, of the 26 patients who received hydroxychloroquine, 6 patients also received azithromycin, and again, that was to prevent bacterial superinfection, and that was added at the discretion of the investigators. 16 patients served as controls, and in the control group, 0 patients were lost to follow-up. So here are the baseline demographics. I won't go over this in detail. It's there for you so you can take a look at it if you'd like. Go ahead and pause the video. But really, the main point is that the groups were pretty equal with the exception of age. So it looks like the group who got treated with hydroxychloroquine were a little bit older than the control group. And based on what we know of risk factors for poor outcomes in COVID-19, that would actually disadvantage the hydroxychloroquine group. So kind of interesting. Again, take a look at that when you have some time. So what was the intervention? It was hydroxychloroquine 200 milligrams 3 times daily for 10 days, plus or minus azithromycin, and that was given in traditional Z-Pak dosing, 500 milligrams on day 1, followed by 250 milligrams per day on days 2 through 5, again, at the investigator's discretion to prevent bacterial superinfection. The primary outcome was SARS-CoV-2 virologic clearance at day 6 post-inclusion or post-enrollment. So they really weren't looking at clinical outcomes. They were just looking at the ability to clear the virus itself. Secondary outcomes included virological clearance during the entire study period instead of just day 6. Clinical follow-up, and so they included in clinical follow-up temperature, respiratory rate, length of stay, and mortality. In the paper, these weren't reported on, so we really don't have a lot of information. All they reported on was virologic clearance. And then finally, their last secondary outcome was adverse events. So, bottom line, are chloroquine and hydroxychloroquine effective treatments for COVID-19? The answer is it's probably too soon to tell. Early results show great promise, and lab results certainly show great promise. However, to date, clinical data is extremely limited. Clearly, additional research is needed to confirm these initial results, and as I mentioned, there are significant limitations to the studies that have available data. So I would be hesitant to say that yes, this is the way to go, but I will say again, the early results show great promise, and I think that promise is why everyone is so excited about hydroxychloroquine and chloroquine for the treatment of COVID-19. So tomorrow we will also have a new episode of 15 on COVID-19, and so our topic for tomorrow will be an overview of COVID-19. We maybe put the cart before the horse a little bit, but we thought it was really important to get this information about chloroquine and hydroxychloroquine and COVID-19 disease out there. Tomorrow we'll circle back and do a more general overview of COVID-19. So, do you have questions about COVID-19? Our goal here at APHA is to provide you with the information you need to educate yourself, your colleagues, and your patients about COVID-19. If you have suggestions about topics you'd like us to cover in future episodes of the 15 on COVID-19 series, please email them to covided@aphanet.org. Again, that's covided, all one word, at aphanet.org. Please note that this is an inbound-only email address. APHA is using this only for the collection of suggestions for future topics. And with that, thank you very much. I hope you enjoyed our inaugural episode. I apologize we went a little over 15 minutes, but hopefully you found the information helpful and can go out and educate your colleagues. Thank you very much, and we hope to see you tomorrow.